Right. Can somebody say something? I'm not hearing anything.
We can hear you.
Right, great. Before we begin, are there any questions? All right, before I share my screen, let me talk a little bit about the grading. And I'm going to call up the grade book to help me talk about the grading. I know I got the last lab graded, which I think was lab 2, but I didn't get it graded until this early this morning, very early this morning. But um, if I had gotten it graded a day earlier, I would have stated that you have to turn it in this Saturday, but I didn't get it graded Wednesday morning. It was Thursday morning, early Thursday morning. So I've decided that you have until next Saturday, not this Saturday, which, let's see if I can remember what my calendar is. So not the 11th, but the 18th. Lab 2 will be turned in again for the second grading. Any questions about any of that? What? That's the wrong one. All right, I also got the extra credit picture assignment graded. Just looking here. I think I got the last lecture assignment graded too. Worksheet for lecture 2 is graded, as well as lecture 1, which I think I told you that last week. I have not graded quiz 1C, especially question 1, or have I graded? I don't think I've graded the quiz 1B either, practice quiz 1B for question number 1. Hopefully, quiz number 1 asked you to show me your scan in question number 1. Can somebody confirm that?
Yes, it did ask us.
All right. Yeah, I'm saying it in question number 1 though.
Oh, yes, it was.
Okay, great. Um, that's the way all the quizzes will be, although I've got to do that for the future quizzes. Move it into question number 1. Um, I think the last quiz has it there already, but every other quiz does not. I've got to move it there. Okay, and I got something else graded. What the heck was it? I've not graded, at least most students, the worksheet for Lab 1. I'll be trying to do that today and tomorrow. I think that's it. Any questions about the grading?
I have a question. On the syllabus, it says we have a test next week. In the grade book, it says it's on the 22nd. Is it on the 15th or the 22nd for quiz 2?
Don't know. Let me take a look here. Quiz 2 is going to cover labs 3 and 4, chapter 3, which we haven't really got started on yet, and the end of chapter 2. That looks like in the syllabus it's wrong, but let me take a look here. And you're asking a difficult question here. That's why it's taking me time to figure this one out. Um, and in the online page, isn't this week 3? Oh, all right. So week 5 is what you're saying it is. I don't have it a link to it to week 5 lecture either. I'm not sure. I'm going to have to get back to you on that. So let me make a note of it, and I'll try and do that after the lab today. I'm going to guess that the syllabus is incorrect, but I'm not positive yet. So let me look into. Quiz 2, right?
Yes, quiz 2. In the grade book, it's not on the Canvas modules, but in the grade book, it says it's due on the 22nd.
All right. Um, like I said, I'll get back to you. This is taking too long, so I'll get back to you on that. Hopefully, I'll have that information for the lecture. All right. Any questions about anything other than obviously quiz 2? If not, let me, oops, that's the wrong thing. Share my screen now. There we go. You should be seeing my screen. Not that there's much here to see, but so today is October 9th, and we are supposed to be working on Lab 4 stains today. Any questions about any of that? Let me put a question mark here. Come on. All right, let's move on to Lab 4, which I think I've got open here. Lab 4. That's an odd. Yep, that's why I got the wrong file open. All right, Lab 4, staining techniques. I know it didn't have it had a name to it. We're going to talk about the simple, the differential, and the special stains. You do need to do some reading before working on the lab. So you need to read about the size and the shape and the arrangement of bacterial cells. Only 2 pages, or maybe even a page and a half. Bacteria cell wall, read about that, page 80 to 83. Read about the simple stain, only a page. Read about differential stains. It's going to be page 65 to 67, of which page 65 to 66 is talking about the gram stain. And then pages 66 to 67 is talking about the acid-fast stain. These are the 2 most common differential stains in microbiology. And then we'll talk about special stains, page 67 to 68, talking about the negative stain for observing cell capsules. And that's page 67 and page 75 to 77. Read about the endospore stain. That's page 68 and page 92 to 94. And then read about the flagella stain. And that's page 68 to page 77. Well, excuse me, page 68 and page 77 and 78. Any question about any of that? All right. There are some supplemental materials in today's lab. There is some video clips and there's a virtual lab. Upon learning this lab, you should be able to prepare a bacterial smear for staining, understand the principles and procedures used when performing a simple stain, a differential stain, and special stain. They each have techniques, and you should know these techniques. And then 3, interpret the results for a simple stain, a differential stain, and for several special stains. Be able to define the following terms. What's a simple stain, a differential stain, a special stain? What's a coccus bacteria? What's a bacillus? What's a sporillum? What is peptidoglycan? What is a gram-positive cell, a gram-negative cell? What is the acid-fast stain? What is a primary stain? What is a mordant? What is a decolorizer? What is a counterstain? What is the cell capsule? What is an endospore? And what are flagella? Any question about any of that? All right. Bacteria are essentially transparent. Therefore, it is necessary to stain them before they can be observed at least clearly under the microscope. When preparing a bacteria smear, you put a drop of sterile water on a clean slide, and then you add just a little bit of bacteria using an inoculating loop. You mix the drop of water and the bacteria to make a smear, and you spread it all over the surface of the slide. You only want a thin layer of cells, so you only want to add a little bit of cells to the slide in making a bacterial smear. You then allow the bacterial smear to air dry, and then the organisms are fixed onto the slide by gentle heating. The heating actually attaches the bacteria to the slide. If they're not air dried and heat fixed, the bacteria wash off by the staining process. Any question about any of that? All right. A simple stain is a technique that uses one dye to stain bacteria cells. You notice how I crossed that basic dye off? It can be an acidic dye, but it is one dye. And we'll talk about acidic and basic dyes in a little bit. You can see the shape and the arrangement of cells in the simple stain, but they all stain the same. A differential stain typically uses two or more dyes to provide additional information about the cell, such as whether the cell is gram-positive or gram-negative. Lastly, special stains are used to stain a particular bacterial structure, such as the cell capsule, cell endospores, and flagella. Any question about any of that? Sorry, there were some lines coming through that one. I'm not even sure where those lines are coming from, but um, so a special stain is a special stain we use to see special structures in the bacterial cell. All right, this graph is showing you the different arrangements, at least some of them, that cocci can take. There's an individual cocci, and that we call a singlet. There are diplococci, and that's not shown here, neither is the singlet. There are tetrads, that's four cocci stuck together. Actually, diplococci is right there, sorry. Didn't notice that. And then there's streptococci, shown here. That's where cocci are in a linear arrangement, one after the other on a string. And then there's staphylococci, where the cocci are stuck together, sort of like a grape cluster. And that's actually what staphylococci means. Uh, Spherical cells, which are cocci, stuck together like grapes. That's what staphylococci means, literally. Besides cocci, bacteria can have a bacilli shape, and there's a bacilli shape there. Bacteria can also be spirilla, and that's the spirilla there. That's sort of like an S-like shape, spirilla. There can be coccobacilli, and that's in between a bacilli and a cocci. And if you take a bacilli with rounded edges and make it smaller and smaller, you will eventually get coccobacilli. Sort of an elongated cocci, or you can call it a truncated bacilli, either way, coccobacilli. There are also some cells which are pleomorphic, that's sort of being shown here. Most of the bacterial species are monomorphic, meaning they have one shape, but some of them are pleomorphic, where the cells take on different shapes. Most of the bacteria species, which are pleomorphic, are bacteria that lack a cell wall. And because they don't have a cell wall, they take on many different shapes. So the cell wall gives the bacteria its shape. Any question about any of that? This here is called the common cellular morphologies of bacteria, meaning the shapes that the bacteria can take. The cells stuck together, that's called a growth arrangement or a cellular growth arrangement, and that's how the cells stick together. All right, let's talk about the differential stains. The first one we're going to talk about is the Gram stain. You don't need to know when it was invented, but it was invented by Hans Christian Gram, and it's named after Dr. Gram. So you should capitalize the Gram stain whenever you write it, because Gram is somebody's name. Any question about any of that? Dr. Gram actually used the Gram stain to differentiate between the bacteria cells infecting a patient and then the patient's own cells. Microbiologists started using his stain, and they discovered that the Gram stain also differentiated among bacteria, separating cells into the gram-positive cells, which stain purple, and the gram-negative cells, which appear pink. And what Dr. Gram did was he was trying to determine the gram-positive bacteria infecting a patient's cells and the patient's own cells stained pink. Any question about any of that? But like I said, bacteria were discovered that they fall into two categories with the gram stain, gram positive, where they stain purple, and gram negative, where they stain pink. And what's happening is the first dye, crystal violet, is being retained by the gram positive cells in the gram stain, and it's being lost by the gram negative cells in the gram stain. Any question about any of that? There's a primary dye. A primary dye is just the first dye or stain used when you're using more than one stain or dye. And you can notice that stain and dye are two words that mean essentially the same thing. So in the gram stain, crystal violet is the primary stain. The Gram reaction tells us about the cell wall composition of the bacteria. The gram-positive bacteria have a structurally different cell wall than the gram-negative bacteria. Shown here, the gram-positive bacteria, this is the plasma membrane here and here, and there's the cell wall of a gram-positive cell, there's the cell wall of a gram-negative cell. The peptidoglycan layer is much thicker on the gram-positive cell, something like 30 layers to several hundred layers, and you can kind of see a few of the layers in this gram stain. There's one there, there's probably one there, there's one there, there's one there, and then it's hard to see. The peptidoglycan layer in the gram-negative cell is much thinner, but it's something like between 3 layers of peptidoglycan and 20 layers of peptidoglycan. And if I blow this up, you can kind of see the layers in the gram-negative cell here. Oops, that didn't work. See if I can get that up. Sorry, that's in the way is the trouble, and I can't shut that down. Wish I could, because I'm not even using it here. What is that called? I don't know how to log out of my account, so see if there's something here. No. Anyone know how to log out of Grammarly, get that icon out of my way here? See, I can't even click that. Let me change the size of this and maybe that'll work. That works. Let's go back to where we were. There, you can kind of see some of the layers of peptidoglycan in here. So there's one, there's two, there's three, four, five, six, seven, something like 7 to 10 layers of peptidoglycan shown here. The point is the peptidoglycan layer is thinner on the gram-negative cells, and the crystal violet dye, when it gets stuck in the peptidoglycan, it can wash out in the decolorizing step. And in the gram-positive cells, the peptidoglycan layer is much thicker, and the crystal violet gets stuck in the peptidoglycan layer. The gram-negative cells also has a lipid layer, and we'll talk about that in a little bit. Any question about any of that? All right. After bacteria have been stained with the crystal violet, Graham's iodine is used as a mordant. A mordant is something that doesn't stain the cells itself. It's not a stain, but it attaches to the primary stain, combines with it, and becomes a bigger molecule, intensifying the stain, making it a little darker, making it less likely that the crystal violet will wash out of gram-positive cells. And so the crystal violet and iodine form a complex in the cell wall. Next, we use alkyl acetone as a decolorizer, and that will pull the crystal violet iodine complex out of the gram-negative cells, but not out of the gram-positive cells, as long as the decolorizer is used correctly. It is simply because the gram-negative cells decolorize more quickly than the gram-positive cells. And that's a warning. If you leave the decolorizer on for too long, you will pull it out, the crystal violet iodine complex, out of the gram-positive cells. But if you don't leave it on long enough, you will not pull at least all of the crystal violet iodine complex out of the gram-negative cell. Did I say that right? If you leave it on for too long, you can pull the crystal violet iodine complex out of the gram-positive cells. And so the decolorizer is a tricky step. It has to be applied for a correct amount of time, and we'll talk about that in this lab. The timing of the decolorizing agent is critical. The bacteria that are gram-negative are now colorless after the crystal violet iodine complex has been removed. And then we stain with a counter stain to color them so that they can show up and you can view them more easily. The counter stain we use is safranin, and that stains the gram-negative cells pink. Any question about any of that? All right. Some cells do not stain well with the gram stain, and sometimes when you stain them, they even look gram variable, where some of them will stain pinkish and some of them will stain purplish. Let me see if I can blow this up again, although it's not terribly critical here. What do you notice about that stain there, that cell there? And where's another one? That cell here, make it the tip of the pointer here, that cell there. What do you notice about these two cells compared to that cell and that cell and that cell? What do you notice?
It's like a little bit of mixed of both.
It's a little bit what?
It's a little bit mixed of both colors.
What colors are you seeing on this cell here?
Pinkish color.
Okay, and what color are you seeing here? On that one there or that one there?
The purple color.
Yeah, that's why it's a mixed variable result because you're seeing one species, and I'll tell you this is one species. Some of them are staining gram positively, some of them are staining gram negatively. But I'm asking you, what do you notice that's different about that cell and this cell compared to that cell and that cell? There is a difference.
Is it the size or the shape of them? They kind of look like the same shape, but maybe the size.
The size isn't totally different. That one is bigger, but that's just because it's a big cell. Um, but that one isn't any different than that
Lighter.
one. Say again?
They seem lighter or like more translucent.
Mm-hmm. That's what I was getting at. This cell here looks like a ghost. So some of the cell, if not all of it, looks like a ghost. You can see through part of the cell even after it's stained. And that one there, kind of ghostly there, or it even looks broken in two or three pieces. What you're seeing is that these cells, the gram negative, are either dead or dying, probably dead, this one at least, and that one. That's why they're ghostly and broken. They're dead cells. And then these are viable cells, and you can see that the boundary of these cells are very distinct and they're much more intense, a complete cell. Whereas that one, the boundary is indistinct and it's not a complete cell. I can blow that up to highlight the boundary, but trouble if I do that. I got to move it away from Grammarly. Yeah, see how the boundary isn't distinct? And it is distinct on that cell and that cell. And then that one is like a ghost, except for this lower portion here. It's a ghostly cell. And that's because the gram negatively staining cells, the pink ones, are probably dead cells. And then the purple ones are probably viable cells. And so they have this distinct boundary to the cell because their cell wall and their cell membrane are still intact. Whereas that cell is clearly a broken cell, so that's a dead cell. And that one's essentially a hollow cell, so it too is a dead cell. That one also is a hollow cell. Whenever I've worked with Mycobacterium, and admittedly I've only used two species, I've noticed that they do stain gram positively. Now the staining will be lighter than the regular gram positive cells because the Mycobacterium had a lot of mycolic acid in their cell wall, and mycolic acid does not allow water or molecules dissolved in water, like the crystal violet, to move into the cell, at least easily. And so the Mycobacterium do take up the purple crystal violet stain, but they do not stain as well as most cells because less of the crystal violet gets inside the cell. Whenever I've seen the Mycobacteria species staining gram variable, I've always asked the student, are these cells 2 days old or younger? Because to do the gram stain, it should be applied to cells that are 2 days old or younger. And if it's older than 2 days old, you should not do the gram stain because the gram stain may not stain correctly. And the students have always told me, oh, these cells, they're 2 weeks old or older. Most of them even over 3 weeks old. And that's why they were staining gram variable, because they had a whole bunch of dead cells in the cell suspension. And so a whole bunch of them were staining pinkish because they were dead cells. The crystal violet was not being retained by the cell wall because the cell wall probably had holes in it. Any question about any of that? So the gram variable result, like I said, I've only seen it. I've never seen it on my own cells because I've never done the gram stain on cells that were older than 2 days old. But on students who they told me that the cells were over 2 weeks old, and it was always Mycobacterium too, meaning a species of Mycobacterium that showed the gram variable. So the Gram variable result is mostly happening, I think, because students are staining cells that are over 2 days old. And the gram stain may not work correctly if the cells are dead. Any question about any of that? All right, let's shrink it down a little and move on. To prevent gram variable staining of the bacteria, avoid using old cells. The gram stain should be done on cells that are 2 days old or younger. And if your cells are older, what you need to do is streak them out and then grow them for a day or two and then stain those cells. You should also prepare a thin smear. If you have a thick layer of cells, one on top of the other, it will be hard getting the crystal violet into the bottom layer of cells if it's gram positive. And then if it's gram negative, it'll be hard washing the crystal violet iodine complex out of the cells. If it's several layers thick, the crystal violet may get stuck in one of the layers of cells. And then you should heat fix the bacteria on the slide only after the smear has air dried. If you heat fix the bacteria while there's still water on the slide, meaning before the slide is air dried, you will cause the water to boil, and that could put holes in the cell wall, which you need intact cell walls in order for the gram stain to work correctly. And so avoid overheating the cells during heat fixing. Excess heat disrupts the cell walls and changes the shape of the cell walls. I've noticed that when anyone ever overheat fixes the cells, it looks like the cells have kind of melted. You should also use fresh staining reagents, and then do not over decolorize or under decolorize. All right, any question about any of that? All right, the acid-fast stain is some bacteria, mainly the family Mycobacterium, the family Nocardia, have a waxy substance called mycolic acid in their cell walls. These bacteria are able to resist acid-alcohol decolorization, so we call them an acid-fast positive bacteria. And that would be species of Mycobacterium and species of Nocardia. We have two acid-fast staining techniques. You don't need to know about the two techniques, but we'll just show you the techniques. The primary stain always uses the stain carbol fuchsin, which is a cherry red color, or you can say a pinkish color. The cells are then decolorized with an acid alcohol, which will pull the acid-fast stain, meaning the carbol fuchsin, from the acid-fast negative cells. You then apply a basic dye to colorize the acid-fast negative cells, and we usually use methylene blue or brilliant green as the counter stain. A counter stain is just the second stain you use, which is a different color from the primary stain. So at the end of the staining process, the acid-fast positive cells, which are always Mycobacterium or Nocardia, will stain differently from the acid-fast negative cells, which are, as far as I know, every other family of bacteria. The acid-fast positive bacteria will stain pinkish, right here, right there, and right there. The acid-fast negative bacteria will take on the color of the counter stain, which will be either blue or green, at least in this lab. So here it looks like it's green because brilliant green was used as the counter stain. Any question about the acid-fast stain? All right. Let's move on and start talking about the special stain. The special stain is a stain we use to stain particular cell structures that's a part of the bacteria to get that structure to show up, because the structure does not normally show up with normal staining procedures. The negative stain is used to stain the glycocalyx. If the glycocalyx is firmly attached to the cell wall and can be viewed under the light microscope, we call it a cell capsule. So there are three types of glycocalyx. We're only going to talk about probably only one here, cell capsule. All right, it's telling me my internet connection is unstable. If you don't hear me, let me know and I'll repeat something. So the cell capsule has molecules in it more than water. You can view the cell capsule under the light microscope, and it's firmly attached to the cell wall. The plasma, oh gosh. Sorry, I'm drawing a blank on what the other type of the glycocalyx is. Oh, it'll come to me. The other glycocalyx, which you cannot see under the light microscope, you have to use the electron microscope to see it. It is mostly water and is very loosely attached to the cell wall. And then in the staining procedure, the structure can easily be washed off of the cells. That's true for the cell capsule as well. You do not see the cell capsule when you use the gram stain because the staining procedure washes the cell capsule off of the cells. Any question about any of that? But even if you use a capsule stain and you have a glycocalyx, which is not a cell capsule, and what the heck is that called? Oh, I almost had it, but I don't have it here. Anyways, it will not show up under the light microscope. The production of capsules can be associated with an organism's virulence or its pathogenicity because the cell capsule protects the bacteria cell from being phagocytized by either a white blood cell or a cell of the immune system. Capsules also change the appearance of the colonies of bacteria. These colonies here look mucoid, and that's because they have a cell capsule. Any question about any of that? I just stopped the share for a moment. Let me look up here something. Come on. Slime layer, that's what it's called. Um, uh, the glycocalyx, which you cannot see under the light microscope, is called a slime layer. Any question about any of that? All right. So a capsule is not easily observed with conventional staining methods because they can be easily washed off. Therefore, we need a very gentle staining technique, such as a negative stain. The negative stain, you just add the cells to the slide, usually they're alive, and then you add the stain, like the the stain here, which is a negative stain caused by India ink. The India ink is very dark. It lightly stains the cells here, so they kind of look gray. And then the cell capsule is clear, unstained by the India ink. This one here is a capsule stain. It probably uses two dyes, one to stain the cell, a dark bluish or greenish bluish color, and then another stain to stain the background, and that looks sort of like a greenish color in the background. And then the capsule is unstained. Any question about any of that? The negative stain typically uses a single dye like the India ink. It could use two dyes though. This one used a dye to stain the cell, another dye to stain the background. Nigrosin can commonly be used as a negative dye in the negative stain. It's a negative stain. You don't need to know that, but India ink is probably the easiest one to remember. All right, endospore staining is a stain that shows you the endospores. Endospores are metabolically inactive, hardy structures that are produced by some bacteria as a defensive strategy to survive unfavorable environmental conditions. Most endospores, or bacteria that produce endospores, belong to the family Bacillus or the family Clostridium. There are two other species that produce endospores, but most of them are in these two families. And I think those other two species each have one species in their entire family. So an endospore is not a reproductive structure, it is a survival structure. When the environment becomes favorable again, the endospore will germinate and grow into the normal bacteria which reproduces and grows. Endospores are not very metabolically active, if they're metabolically active at all, and that differs from the vegetative state, which is very metabolically active, and then the vegetative state can grow and reproduce. This table that I'm showing you here is some of the pathogenic endospore-forming gram-positive bacilli, meaning the gram-positive pathogens that produce endospores of the family Bacillus and Clostridium. And you'll need that table for some of the questions in the worksheet. The Schaefer-Fulton stain, also called the endospore stain, uses malachite green as the primary stain. It initially stains both vegetative cells and endospores. Heat is then applied as a mordant to help the stain enter the endospore. The cells are then decolorized with water, which removes the malachite green from the vegetative cells, but does not remove it from the endospores. Saffron is then applied as the counter stain to stain any cells that have been decolorized. The vegetative or parental cells then appear pink in the endospore stain, and the endospores appear dark green. Let me blow this up so you can see it a little better. Come on. It happened to be in the same part, but shoot. For some reason, Grammarly is now moving wherever I move this. I have to figure out how to close Grammarly. Ah. Quickly get it. Yep, I got one time in there. Shoot. Nope, can't get that out of here. All right, I'm not going to bother anymore. It's going to be permanently a problem. Um, there's green endospore. There is a pink vegetative cell or a perianthal cell. There's another perianthal cell. And here, I'd really like to blow that up. There is a perianthal cell with an endospore inside it. So endospores initially grow inside the parent cell, and then the parent cell dies, releasing the endospore. Any question about endospores? Sorry, I can't blow that up, but Grammarly is going to be a permanent problem. All right, so the endospore is one primary special stain. There's also a flagella stain. Flagella are long thread-like appendages that are attached to the uh, cell for providing motility. So if the cell can move, locomote, or swim, it has a flagella. We're talking about bacteria cell. The only way bacteria cells can move is by a flagella. If we're talking about eukaryote cells, they can move by three different means, one of them being a flagella, and I'm not going to talk about the other ways that eukaryotes can move, at least not in this lab. Flagella is composed of the protein flagellin. The flagella has a particular arrangement on the cell. They can be monotrichous, where there's one flagella on the end of the cell. They can be lophotrichous, where there's multiple flagella on one end of the cell. They can be amphitrichous, where there's multiple flagella, on both ends of the cell. And they can be peritrichous, where flagella are all over the cell. Any question about any of that? You do not need to know the flagella arrangement for any quiz, but you will need to answer it for the worksheet on this lab. Any questions about any of that? Because flagella are too thin to be seen using standard techniques, special stains are required to view them under the light microscope. Like one flagella stain, what it does is you simply use it to coat the flagellum, and then you reuse the stain multiple times until you get many, layers of the stain on the flagella. Is that clear to everyone? I'd really love to blow this one up, but I don't think I'm going to be able to. Oh, what do you know? It didn't follow me. I was able to do it this time. Let's go way up. Large. All right. Whenever you see cell stain with the flagella stain, you'll note that some of them have multiple flagella everywhere. They're peritrichous. Others of them, well, maybe that one there is, no, that's more like a lophotrichus. Let me see if I can find an amphitrichus cell. This is one species in here. In this, that's also lophotrichus. If I look closely, I can find an amphitrichus, but I'm not finding one here. At least now. Anyone see flagella on both ends of the cell? Hard to tell on that one because I can't see the flagellum. Maybe here on this one. And then there's one in here that only has one. Like there's probably one. It looks like about the only one I can find. Oh, let's look for one that has none. Maybe that one there has no flagella. So in something like that, what do you do? What do you call it? Do you call them peritrichous, amphotrichous, lophotrichous, monotrichous, or do you call them all of them? And then I guess there's also no trichous, meaning no flagella. I did show you one. What you do is you call it by the highest flagella arrangement. So this one would be peritrichous, because these here are definitely peritrichous. I think that one's peritrichous as well. And there are other arrangements, but when the cell first divides, it will not have a flagella. And then the cell will make a flagella, and the first one it makes, you'll have a monotrichous arrangement of flagella. And then you'll have a lophotrichous arrangement of flagella. So that should have been flagellum, flagella. And then it'll grow flagella on both ends, so it'll be amphitrichous. And then it will grow flagella all over the cell, so it'll be peritrichous. So this species is peritrichous, and the ones that we see that are less than peritrichous, they just haven't grown enough flagella yet. And given time, all the cells will eventually become peritrichous, at least if the cell lives long enough. Obviously, if it dies before it becomes peritrichous, it won't ever make peritrichous. But my point is the species is peritrichous, but initially when the cell divides, the cell will have no flagellum. And then it'll grow flagella, becoming higher and higher. You also have some species where the highest one can be amphitrichous, so it never becomes peritrichous. And then you have some species of bacteria that only become lophotrichous, and some species that only become monotrichous. And then we have many species of bacteria that have no flagella. Any question about any of that? All right, so that's how you call the species of the highest order of the flagella arrangement. So special stains are required to view them under the light microscope just because the flagella is too thin to be seen with the standard staining technique. You'll never find the flagella in the gram stain. It just is too thin to be seen. All right, that's it for the special stain. Here's the terms for this lab. You should know these terms. References you don't need to view. We have a little video on how to prepare a microscope slide of bacteria. And let me mention that when you make a bacterial smear, the bacterial smear is a separate preparation than staining the cells. So whenever you stain cells, you first make a bacterial smear, but then the staining procedure occurs, and that's a separate protocol. So we have the protocol for making a bacterial smear, and you don't have to ever stain that bacterial smear. You could view it under the light microscope without any stain, or you could do something else with it. My point is the bacterial smear is a separate step, a separate protocol than and an independent protocol than the staining protocol. So we have a little video on making a bacterial smear. A little video on gram staining technique, and then the video on physiology of bacteria. Let's go to the worksheet. I think that's what I want to go to next. I'm going to make sure the worksheet covers it. You're not going to be physically performing any of these activities in laboratory 4, but you must understand how to prepare a bacterial smear for staining, understand the principles and procedures used in the various staining techniques, and be able to interpret those results using a microscope. Now, like I said, remember a bacterial smear is a separate protocol than the staining. Usually, we do a bacterial smear before we do the staining. But you could do a bacterial smear and never stain it. And then you can also stain cells without doing a bacterial smear. Any question about any of that? All right. So in procedure 1, we're going to prepare a smear for staining. We have a little video clip on making a bacterial smear, and then a little video on air drying and heat fixing the bacterial smear. You should review the steps of making a bacterial smear. You first put a drop of water on the slide, usually sterile water because you don't want bacteria being added by the water. And then mix a small amount, just a very tiny amount of bacteria in the drop of water. Mix it in, and then spread the drop of water all over the slide. Let it air dry. Heat fix the bacteria onto the slide. That'll stick the bacterium to the glass of the slide. Let the slide cool. That's usually just a few seconds. And that's it for the bacterial smear. You can now do the staining. The staining is a separate protocol, and I'm telling you this time and time again because students seem to get mixed up between making a bacterial smear and then doing the staining. They're separate and independent procedures. And then the first question in procedure 1, I've got the questions always with a question mark, is what are 4 common errors when preparing a bacterial smear for staining? So list me 4 different errors that you can make in making a bacterial smear. And then we have procedure 2 on simple staining. We have a little video clip on making a simple stain, so watch that video clip. When you're doing a simple stain, you just add 1 basic dye, meaning a basic dye. You could actually use a negative dye, but here we're going to use a basic dye. Usually methylene blue or crystal violet. And you obviously are going to add the dye to a bacterial smear. So you do the bacterial smear first, and then you do the staining. After about 1 minute of the stain on the cells, you then remove the stain. Usually you wash it off with water, and then you can blot it dry. Blotting paper is very thick paper that's almost like cardboard, and you rip off a piece of it. Usually I like to fold it. I don't know if you can see this. Put the slide in the blot paper and then press it down. You do not want to move the blot paper against the slide because that'll remove the cells, or move the slide against the blot paper. You just press down the blot paper. That gets rid of the most of the water, enough of the water that you can view the cells under the microscope, and you can even use oil on the slide. Enough of the water has been removed that you can even use oil. And then you view the slide under the microscope. And that's what a simple stain is. Recall that all the cells will have the same color, so you have to differentiate the cells on their shape and on maybe their cellular growth characteristics. Let me tell you, the questions ask you, E. coli, what's the cell morphology of E. coli? It's not a spirillum. The reason why it kind of looks like a spirillum is the cells are stuck together. Like that's 1 cell, and actually that might be 3 cells stuck together right there. And we're looking at E. coli and Staphylococcus aureus. So what's the shell morphology? Cellular morphology, not the cellular growth arrangement of E. coli and Staphylococcus aureus. So that's procedure 2A. And then procedure 2B, what is the total magnification? And that is given in the worksheet. Although you might have to calculate it, you're given the information to do so. All right, procedure 3 is doing the gram stain. We have a little video clip on doing the gram stain procedure. We also have a practice performing the gram stain by using a virtual lab. Let me open that now. It may take a little while to load. All right. The gram stain. Hit continue here.
Hi, Lab Assistant. Welcome to the yogurt section of the dairy processing plant. It appears that our latest batch of yogurt might have been contaminated by...
I'm not going to listen to it now. I moved through it quickly. You can read through or listen to it. I'm just going to move to the steps I want to talk about. You kind of need to know about this, but we're going to skip it. I'm just going to get directly to the procedure. All right. In this lab, the arrow tells you
Bring the striker to the Bunsen burner with the flint side facing down and squeeze the striker to create sparks.
So what you do is you take your mouse on where the arrow is starting to move what you're doing down, and then just there it'll strike itself.
We need to dilute the yogurt before we put it on the...
I'm going to skip this. And once again, the arrow tells you what to do. You take the pipette, bring it down into the solution. Move it over, heat it until it's red hot. Let it cool. Move this down to pick up the diluted yogurt. Put the diluted yogurt in the solution, mix it up. Let's just move on. Get to the gram stain. Come on. This is actually incorrect. You don't want to heat fix the bacteria when it has water on it. You should let the slide air dry. All right, here's the gram stain. So let me go ahead and turn on the
under slow running water in between each chemical. The first is crystal violet. This is our purple stain, which will indicate gram-positive bacteria. Using the dropper, squeeze out enough drops to cover your sample. Let that sit for 60 seconds. Now rinse it under slow running water. Now use the bottle of Grams iodine. Grams iodine will fix the dye to the cell walls of the bacteria and make it permanent. Squeeze several drops onto the sample, enough to cover. Let that sit for 60 seconds. Now, rinse it under slow running water. Now we are going to use the alcohol solution. This will wash off the excess. Use the alcohol dropper to cover the sample in alcohol. Let the alcohol sit for 30 seconds. Time is very important in this step. Don't let the alcohol sit for more than 30 seconds.
All right. This is 95% alcohol being the decolorizer. Usually we use acetone alcohol mixture, in which case you only let it sit for about 5 seconds. So it does depend on what your decolorizer is. This one is using 95% alcohol, and so we're letting it sit for 30 seconds because this decolorizer is not as strong as the acetone alcohol decolorizer.
Now, rinse it off under slow running water. You now have stained the gram-positive bacteria purple.
Okay.
The next.
All right. So that goes through the gram staining procedure. I just wanted to show you how to do that, and this will go through the rest of the gram stain. Any questions about that little tutorial? Virtual lab? Like I said, the virtual lab uses a different decolorizing solution than we will discuss. So this step is longer in the virtual lab than the decolorizing step that we will talk about. This is showing you a review of the steps for performing the gram stain. First, you stain the cells with crystal violet. All of the cells will stain purple from the crystal violet. And this is a bluish purple color. Both the gram-positive and the gram-negative cells will stain bluish purple. You then add the mordant grams iodine onto the slide. The color is a little bit more of a purple from a blue purple color. And the cells also stain a little bit more intensely. And that's because the iodine combines with the crystal violet to make a larger molecule in the cells. However, usually when students are observing the slide, they cannot notice the intensity because it's slight, but there is a stronger staining intensity after you add the iodine. As well as the color is slightly different. It's more of a purple color from the blue purple color, and that's not shown here. You then, after washing the solution off, you then add the alcohol acetone wash. This is the colorizing step, and this is done for only about 5 seconds. It's best to add your decolorizer. Pretend this is your slide. You add your decolorizer while you're watching it, and when the blue stops running on the slide, you stop adding the decolorizer, and that will happen in about 5 seconds if it's the acetone alcohol decolorizer. Any question about that? The decolorizer will pull out the crystal violet iodine complex from the gram-negative cells, but not from the gram-positive cells, at least if you do the decolorizing step correctly. Any question about any of that? So the gram-negative cells here will be clear, and then the gram-positive cells will be purple. You will then immediately water wash the decolorizer off, because even the trace amounts of decolorizer that are left on the slide will continue working, and given enough time, they will pull the crystal violet iodine complex out of the gram-positive cells. So after adding the decolorizer for the correct amount of time, which is usually the best way to do it, is to simply watch when the decolorizer stops running the blue on the slide, which will be about 5 seconds. And then you wash immediately to remove the traces of the decolorizer from the cells. And then you stain with safranin, the counter stain, to add color to the gram-negative cells. And they will become pinkish, and then the gram-positive cells will retain the purple color. Any question about any of that? All right, here is the E. coli, and that shape actually looks correct. Give me the gram reaction of gram E. coli, and then give me the cellular morphology of E. coli, meaning the shape, whether it's coccus or bacillus, and then give me the gram reaction of Staphylococcus aureus, shown here, and give me the cellular morphology, the shape of Staphylococcus aureus. For procedure 3, also answer question 3A, 3B, 3C, 3D, 3E, and 3F. Staphylococcus aureus, give me the color, pink, purple, or colorless, after you apply the primary stain. 3B, E. coli, after the primary stain is added, give me the color. 3C, Staphylococcus aureus, after the acid alcohol acetone step, meaning the decolorizing step. And then 3D, E. coli after the alcohol acetone step. 3E, Staphylococcus aureus after the counter stain is added. 3F, the color of E. coli after the counter stain is added. Any question about any of that? All right. For procedure 4, we're looking at the acid-fast stain. We have a little video clip on the acid-fast stain, so watch that video. And then review the steps of performing the acid-fast stain. We're going to use here the modified Kenyon acid-fast bacteria stain. First, you add carbol fuchsin. It stains all cells red or pinkish. It is the primary stain. You then apply there might be a mordant in there. You then apply the decolorizer. After you apply the decolorizer, the acid-fast negative cells will be unstained, meaning they will not retain the carbol fuchsin. And then the acid-fast positive bacteria will retain the carbol fuchsin, so they'll remain pink. You then apply a counter stain. Here we're using brilliant green, and the acid-fast positive cells will stain pinkish or reddish, and the acid-fast negative cells will stain greenish. Any question about any of that? After each step in the acid-fast stain, you gently rinse the slide with water. And I forgot to mention that in the each step in the gram stain, you also wash the slide with water. If I ever ask you for the steps for the gram stain, for example, you only need to tell me in one place, either the beginning or the end, that after each step, you wash the cells with water, gently wash with cold water. You don't need to say, I apply the crystal violet, then I wash with water, then I apply the grams iodine, and then I wash with water. You don't need to say that washing with water each time. Someplace you just need to state, either at the beginning or the end, that after each step, I wash with water. All right, any question about procedure 3? Or procedure 4, I guess? So the questions of procedure 4 are these two smears that were stained with an acid-fast stain. All the organisms denoted by an arrow are either AFB positive, meaning acid-fast positive, or acid-fast negative. Supply an answer for each arrow. So on this one here, you can say the bottom arrow is either acid-fast negative or acid-fast positive. You can write it there, or you can write it either above the picture or below the picture. But you need to answer each arrow, whether it's acid-fast positive or acid-fast negative. And then do that also for this picture here having 2 arrows. And most people on the second one say the leftmost arrow and the rightmost arrow. Any question about procedure 4 and the questions? So don't forget the questions. It's the arrows. If you don't get the arrows answered, it'll be a -4. Minus 0.4, sorry. Special stains. We have a video clip on performing the endospore stain and a video clip on performing the negative stain, looking for a cell capsule. And then use these 3 bacterial smears that have been stained at 1000x total magnification for answering the question. So 5A goes to picture A, 5B goes to picture B, 5C goes to picture C. So 5A says this is the Schaefer-Fulton stain, which uses which stain as the primary stain, and it ultimately dyes which cell structure, which are produced by some bacteria to aid in survival. Any questions about any of that? So you have to interpret what you do for filling in the blank. But this is talking about the first 5A1 is going to be which is the primary stain. So give me the name of the primary stain. And it dyes which cell structure. And it tells you the cell structure is produced by some bacteria to aid in their survival. And you can see the structure right there in that figure A. And then what are some examples of pathogenic endospore producing bacteria? So you must write 2 or more examples of pathogens that produce endospores. If you only give one, it'll be partly wrong because it does ask for examples. So that's plural. So 5B refers to question figure of 5B. And this is what stain that coats long whip-like appendages which aid the cell in motility. So give me the name of the stain. And then state the flagella arrangement type. The number of flagella that this species has. Right there. And then for 5B3, what are some examples of clinically significant bacteria that produce flagella? Once again, you must do more than one, meaning you must do at least 2. And then 5C, referring to figure C, 5C, this is which stain that uses India ink or nigrosin and can be used to detect the presence of what cell structure, which is located exterior to the cell wall and prevents phagocytosis. Any question about 5A, 5C1, and 5C2? And then what are some pathogenic microorganisms that produce capsules? Once again, you must give an answer more than one answer, more than one species. All right, then do 2 case studies. Case study 1, a 45-year-old female is experiencing some respiratory issues. Based on her clinical presentation, the doctor orders a chest x-ray, shown here. And then they take her sputum, shown here, for acid-fast bacteria culture. So an acid-fast bacterial smear is made and stained, and that's shown here. And based on the findings below, what is the most probable diagnosis? You must name the species which is causing the infection. So in the x-rays, this is an irregular x-ray. The x-ray should be dark like that, and this region is not dark, and this whole region is not dark. So something's inside the lungs. And there's the stain, and that's actually showing you the species of bacteria causing the infection, and you must name the species. Don't give me the disease. It'll be partly wrong if you give me the disease. Case study 2. A 64-year-old diabetic patient is admitted to the hospital with soft tissue necrosis, shown here. A routine culture and gram stain is ordered. The bacteria grew aerobically. Based on the findings below, what is the most probable diagnosis? You must name the species which is causing the infection. So there's the clinical presentation. There's the gram stain of the bacteria. and this is the Schaefer-Fulton stain. So name the bacteria that's most likely to cause this infection. And you can find that, by the way, in the table in the lab module. Any question about any of this? Oh, on the case studies, you could go ahead and put this information into an AI, and it will give you the answer. I'm not sure how you could do the pictures, but you could probably describe the pictures. If you do that, you'll get a quick, easy answer, especially for this one, but then you will not practice doing a case study, which you will have on the final, maybe on a quiz or two as well. So you should do the case studies on your own. And then if you wish to check your answer, you could check it with AI or with another student. But like I said, if you use AI to get your answer, you will not be able to answer the case studies on the final, and there will be several. Any questions about any of that? And another thing, if you give me a disease which is not discussed or a bacteria which is not discussed in this lab module, it will be wrong. So if you use AI and you get an answer, which is an answer that this lab module, a species which this lab module is not talking about, it is wrong. This is a bacteria this lab module is talking about. Any questions about any of that? All right, go ahead and get started on the lab. I will be here until 4:30 to help you answer any questions in the lab worksheet or any other questions. But if you have a question, make sure you ask it before the last student logs out, because once the last student logs out, I'm going to log out as well. All right, any questions? All right.